mainly three reagents is very important for carrying out the acid fast staining. First one is Ziehl-Neelsen carbol fuchsin. Ziehl-Neelsen, the person who was discovered, and the reagent name is carbol fuchsin. And second one is acid alcohol, and third one is methylene blue, right? So these are the reagents which is packed, which is filled in this container. We need to use these reagents in the procedure staining. So we have named the Ziehl-Neelsen carbol fuchsin as primary stain because this is the first stain which is used in this staining procedure. So it is regarded as the primary staining. And second one is acid alcohol. So acid alcohol, in the sense it is a combination of both acid and alcohol. So it contains 3% of HCl and along with the 95% of ethanol. And coming to the methylene blue, it acts as counterstain or secondary stain, and which contains 3% of methylene blue, right? So these three reagents are used for the acid fast staining. So coming to the principle, so just noted acid fast bacteria has high lipid content, which is the high mycolic acid content in their cell wall. That high content cell wall which retains the red color of the primary stain. So primary stain is nothing but the carbol fuchsin stain. So carbol fuchsin is a red color, so that acid fast bacteria retains the red color of the primary stain. So Mycobacterium species, that is the acid fast bacterium species, retains the color of the carbol fuchsin even after the treatment with acid alcohol. Okay, so if even after adding the acid alcohol decolorizer in that cell wall, the microorganism, the acid fast bacteria, will not able to lose the red color. Okay, so it retains the red color fully even after adding the hydrochloric acid and ethanol. So in contrast to this, non-acid fast bacteria lacks the large amount of lipid content. So it loses the primary stain, it loses the color of the primary stain, and it just decolorizes all the red color from the bacterial cell wall. So coming to the procedure, just note it down. You want to take a clean and grease-free glass slide, because this staining procedure is performed in the form of glass slide. So take a glass slide in which you need to prepare a bacterial smear. So bacteria, so it should be a fresh culture, because fresh culture has the ability to grow and reproduce newly. So for staining purpose, we need to add the fresh bacterial culture media. So in that glass slide, we need to prepare a smear, bacterial smear, with the help of inoculation dope. And further preparing the bacterial smear, we need to add the carbol fuchsin dye. Okay, so after preparing bacterial smear, we need to pour the carbol fuchsin 1 or 2 drops in that bacterial smear, and it is just go to the heat fixation, because heat fixation process helps the bacteria to absorb all the carbol fuchsin color in its cell wall. Okay, so it is fixed with carbol fuchsin, and after adding carbol fuchsin, we need to heat fix the slide for proper absorption of red color carbol fuchsin dye, and wait for 1 minute, and after 1 minute it just rinse with the water. Okay, so the bacterial glass slide is there, we just rinse the slide and rinse the bacterial smear with the water slightly, and further adding water, we need to add the acid alcohol reagent drop by drop in that bacterial smear, so that excess of the red color carbol fuchsin dye will remove off from the non-acid fast bacteria, whereas acid fast bacteria, which does not remove the carbol fuchsin dye because it has a very thick layer of mycolic acid in its wall. It retains fully the red color. And after adding acid alcohol, wait for 1 minute, and again it is rinsed with water. And finally we want to add the counterstain. So counterstain is nothing but the methylene blue stain. So after adding methylene blue stain, we need to wait for 1 minute, and after 1 minute it should be rinsed with water. Now all the excess red color in the non-acid fast bacteria will remove, and the methylene blue will stick into the non-acid fast bacteria. So after adding methylene blue, we just rinse it with water and air dry the slate, and coming to view under the microscope, you could see the acid fast bacteria in red color and non-acid fast bacteria in blue color. Okay. So this is the step we need to add. We want to take a slide and add the carbol fuchsin, which is nothing but the primary stain, and application of heat, it acts as mordant. And after rinsing with water, again we want to add acid alcohol, which is nothing but decolorizer, and wait for 1 minute, and again rinse with water, and again we want to add finally methylene blue color in its slide. So you could see the acid fast bacteria in red color and non-acid fast bacteria in blue color. So this is the overall interpretation. So you could see the acid fast bacteria was appear in red color, which is rod shape or bacilli shape, and non-acid fast bacteria will appear as blue color, which is in cocci, round shape. Okay. So this is the main interpretation. You could remember acid fast bacteria having red color rods and non-acid fast bacteria having blue color cocci. So this is the exact picture. This is the diagrammatic representation where we viewed under the microscope. So coming to the acid fast versus non-acid fast bacteria. So acid fast bacteria, which retains the red color of the carbol fuchsin after treatment with acid alcohol, while the non-acid fast bacteria will lose the red color after adding acid alcohol, and it retains the blue color of the methylene blue. So acid fast bacteria appear as red color, whereas non-acid fast bacteria will appear as blue color. So that I have mentioned in the color: acid fast red color, non-acid fast blue color. And coming to the another difference, acid fast bacteria, which takes a very long time to grow, that is 3 days to months. Okay, so acid fast bacteria in the sense we could say the mycobacterium as a very good example. So that mycobacteria will take a very longer time to grow, that is around 3 to 30 days. And non-acid fast bacteria, which grows very fast, that is within 24 hours they can able to grow and multiply and reproduce. So a very good example of non-acid fast bacteria is E. coli and Staphylococcus aureus. So if we cultured the E. coli and Staphylococcus aureus, so the bacteria can able to grow at 37 degrees Celsius for 24 hours. Within 24 hours they can able to grow, multiply and reproduce. But the acid fast bacteria will take a longer time to grow, that is to grow about 3 days to 3 months it has the ability to grow. So that's why we are caused the tuberculosis at maximum at least 1 or 2 months the symptoms is there, we could confirm the tuberculosis attack. Not by a single cough or cold we need to declare that it is tuberculosis. So the growth of the bacteria will differs from the acid fast bacteria and non-acid fast bacteria. You could clearly seen in this difference. So these are all about the acid fast bacteria, non-acid fast bacteria, and these are all about the acid fast staining. So we can conclude that the acid fast bacteria is discovered by the Paul Eldridge, and later it was modified by the Ziehl-Neelsen, and it is mainly used to differentiate acid-fast bacteria and non-acid-fast bacteria. And this having the three reagents like Ziehl-Neelsen, carbol, fuchsin, acid alcohol and methylene blue. And you could see the principle, we have seen the procedure, and we have seen the interpretation, and finally we have concluded the difference between the acid-fast bacteria and non-acid-fast bacteria. So thank you all. We can see the next class with the another interesting topic. Thank you so much.